Hello students, today we will discuss about the petrological microscope which is being used to study the slides of rocks and minerals in order to identify the minerals and the rocks. So let us start. The petrological microscope is otherwise called as a refractive microscope, or it can be called as polariscope. So it is a device to magnify the image of the object so that the micro objects can be seen perfectly and can be identified. So, a petrological microscope is used to study and identify minerals and rocks in thin section through transmitted light. We use transmitted light to study the rocks and minerals, but we use reflected light to identify the ore minerals which are opaque. So, here we will discuss about the refractive microscope. It is an essential tool in petrography which involves careful description of rock composition, textures and microstructures. So, petrography is one important component of rock identification, and that can be possible if we identify the rocks under petrological microscope. There are different manufacturers of petrological microscopes such as Nikon, Zeiss, Leach. Each of them vary in different aspects, but fundamentally they have same design and construction. Following are some of the essential components of petrological microscope. First is the illuminator or light source which occurs at the base. Then over it we have the substage assembly that contains lower helical or polarizer, condensing lens for orthoscopic illumination, and the auxiliary condensing lens for conoscopic illumination, and it is followed by aperture or diaphragm. So besides this, we have above it the superstage components above the microscope stage. So we have above the substage assembly we have the microscope stage, then it is followed by superstage assembly that includes objective lens, the slot, the oponical, button lens, ocular eyepiece, and these are the parts. And after discussion of all these parts, we will see the processing mechanism and the direction convention. But this here is the simplest model of a petrological microscope. We have the base, this is base, then mirror to allow the light into the microscope, then polarizer, condensing lens, diaphragm, this is stage, objective, analyzer, button lens, eyepiece. So these are the important components that have been shown in a simplified form in the petrological microscope. Now let us discuss about these different parts. First is illuminator. All microscopes have a flat base that contains a light source or illuminator. It may be daylight that is sent up by rotatable mirror fixed at the base of the microscope stage, which has one plane side for uniform illumination and a concave side for more illumination. This is the case when we talk about simple petrological microscopes, which have the device of sending the light into the microscope through a mirror which is fixed at the base of the stage. It is having a concave side and it is having a convex side. So light falls over it and it is being sent up in this manner. But this is rotatable and we can rotate it and allow the light into the stage. Besides that, in good microscopes we have a power source having an incandescent ball that supplies the light which is directed upwards with the combination of mirrors and lenses that get filtered through blue glass to provide color sensation approximately to that of sunlight. Many models have rheostat control that permits the intensity of light source to be adjusted, to be set in adjusted condition. Now we will discuss about the soft stage assembly, the components which are fitted below the stage. So among them is the polarizer or lower nickel. It is a device to produce polarized light out of ordinary light, which is directed upwards to the stage with the vibrations taking place in one plane, either parallel to east to west or north south direction of the microscopes. So the very function of the polarizer is to produce polarized light and supply it to the microscope. In many microscopes, there is a rotation facility to adjust the direction of vibration of polarized light. And after this polarizer, we have the condenser. There are two types of condenser. One is orthoscopic condenser, the other is conoscopic condenser. So orthoscopic condenser, it is a fixed type of condensing lens that focuses light on the area of the sample by a set of parallel light rays through orthoscopic illumination. So this orthoscopic illumination is being used to study different optical characters of minerals, particularly the habit, color and pleochroism, refractive index, fractures, twinkling effect, cleavage, alteration. These are some of the properties which are being studied by orthoscopic illumination, where convergent polarized light is not required. But when we talk about conoscopic condenser, it provides a cone of light rays, or we provide convergent light source. We provide convergent light source. In this manner, light falls over the slide of the mineral and the properties are being studied through interference figure. So to produce interference figure, we use conoscopic condenser. For the studies of properties like color, pleochroism, refractive index, fractures, twinkling effect, cleavages, alterations, these properties are being studied in orthoscopic illumination. And the microscopes have the provision either to swing in or swing out the cornoscopic condenser into the field of view. Then we have above or within the condensers, within the two condensers, we have the iris diaphragm that is used to control the amount of light into the microscope, or in order to regulate the size of the cone of light so as to have a contrast in the image seen through the microscope. It is not intended to be used to adjust the intensity of the illumination, just to control, regulate the size of the cone of light rays. So these are the four components that are used below the stage. Above the stage or above this sub-stage assembly, we have the microscopic stage, which is circular, rotatable, and it has a hole in its middle for keeping the slide. For keeping the slide, over this slide is being placed. Which is placed for the identification of the rock and mineral. And there is circular scale on the stage which reads from 0 to 360 degree, along with a small vernier scale attached here, which are used to measure some of the angles, particularly the extinction angle, keeping the mineral away from its extinction position. In highly illuminated condition, or to study the interference color in 45 degree position from extinction. So the stage has that facility to be rotated, and by this process it is possible to find out the extinction angle of minerals, vibration direction of the minerals, 45 degree position from the extinction position. For that we use this circular scale, also this small vernier scale. Then we have the super stage assembly. It includes the following parts: objective lens, then we have the slot, over it we have the slot for analyzer, Over that we have the Bertrand lens, and on the top we have the eyepiece. So what is objective lens? It is a lens to simply magnify the image, to magnify the image of the object, and for that it has certain power. The powers are in this range, 2.5 to 4 or 8 to 10 or 40 to 60, like this. And this is mounted to the circular nose piece that can be rotated to change the objective lens depending upon requirement. Generally, larger the diameter of the objective, smaller is the power, and larger is the free working distance. What is free working distance? It is the distance between the end of the objective lens and the top of the sample. So generally we use the low power objective to study the general properties of minerals. On the outside body of the objective lens, there are markings that specify the magnification, lens type, numerical aperture, cover slip thickness, and tube length, as shown in the diagram below. Here 40 is the power, magnification power. That means it will magnify the image 40 times. PLR lens type, 0.65 is the numerical aperture, 160 is the tube length, then 0.17 is the cover slip thickness. So the numerical aperture of the lens, what is Na, is the measure of the size of the cone of light that it can accommodate. It is given by equation Na is equal to n sin A by 2, where n is the medium between the objective and the slide. Na numerical aperture, n is the refractance of the medium. Generally it is 1 because air, and A is the angular aperture. So this is the angular aperture, and this is the free working distance, slide and objective, tip of the objective. So this distance is the free working distance. So greater the diameter, lower the power, higher is the free working distance. Higher the power, the free working distance is very low. So in that case we have to be cautious when making up and down movement of this objective through the knob. We should be careful so that this tip should not touch the slide, otherwise it may cause harm to this tip portion of the objective. So while if we are using high power objective, in that case we have to be careful. If we are using low power objective, then free working distance will be more, so it will be easy to identify the properties without any precaution. So above the objective we have, in the microscope tube we have, here is the objective, here is a slot position to insert the accessory plates. Accessory plates are being inserted in the slot. Slot is there in the slot through which accessory plates are being inserted to study some of the optical properties. So it is actually in 45 degree position to the vibration direction of the polars. So it is placed above the objective with the microscope tube, and for insertion of accessory plate are compensators in order to identify vibration direction of the minerals, order of interference color, sign of elongation, fast along negative, slow along positive, which is the sign of elongation or the optic sign. The compensators are generally quartz wire, gypsum plate and mica plate. These are the common compensators. Then above it we have the provision for use of this analyzer, or we have the slot where analyzer is fixed. It is another kind of nickel prism or a polarizing device whose vibration direction is perpendicular to that of the lower nickel or polarizer. And if the polarizer vibration is east-west, then another vibration direction will be north-south or vice versa. So that when both are in use, the microscope is said to be in crossed nickel position. In that condition, some of the typical properties of minerals are being studied, such as isotropism or anisotropism, then interference color, extinction, birefringence, then twinning. These are some of the important properties which are studied after the use of analyzer. So when there is in crossed condition, if there is nothing on the stage of the microscope and analyzer is inserted, no light passes through and there is darkness. It results due to the fact that light coming from the polarizer is obstructed by the analyzer, as its vibration direction is perpendicular to that of the polarizer. It is very often seen that there is no perfect extinction shown by the minerals. That happens when we are not aware about the fact that the lower nickel and upper nickel, they are not in perfect perpendicular condition, because through the process of rotation we miss that situation, we miss that condition when the analyzer vibration direction is perfectly perpendicular to the polarizer vibration direction, because polarizer is rotatable, analyzer is sometimes found to be in rotatable condition. So we should ensure, before the study of the rocks and minerals, we should ensure that analyzer and polarizer vibration direction, they are mutually perpendicular or in crossed condition, so that the optical properties can be studied. If they are not in crossed condition, we cannot get perfect extinction or perfect interference colors. Then we have the button lens. Above the analyzer, we have the button lens, which can be pushed into use or can be pushed out of use along the path of the light, and it is generally used to study interference figure in magnified form along with eyepiece in conoscopic arrangement. So button lens is used during conoscopic arrangement for study of interference figure. Then on the top we have the eyepiece. This eyepiece, it is having certain power, 5 times, 12 times or 8 to 10 times. So objective has certain power, let us say it is 40 times. objective. Let us say 5 times eyepiece power. Then total magnification will be 40 into 5, 200 times. So the magnification will be 200 times. So objective and eyepiece, these are the two lenses which are used to magnify the size of the object on the stage in form of image that is identified by our naked eye. So most of the eyepieces contain reticle markings like this. So this is, we say, crosshair. This is useful to center the object on the stage or to keep a mineral in the center of the stage for optical study, perfect optical study. So these reticle markings are there, east to west, north south markings are there, parallel to the vibration direction of the polarizer and analyzer. Also, in some cases, we have the crosshairs are used to center the feature of the interest in the field of view. In some microscopes, eyepiece contain micrometer scale to measure distance between and sizes of the objects on the sample under study. So we have the sample on the stage. So different features which are present in this slide, their size, length, breadth, that can be identified by this micrometer scale. These scales are calibrated with the stage micrometer for each respective objective. Objective wise, this scale has been calibrated. The stage micrometer is a microscope slide on which a millimeter scale is found to be etched. Thus, comparison of micrometer scale and that of the stage micrometer is made to measure the size of the grains in the sample. So this type of scales are seen through the eyepiece. This is ocular micrometer scale. This is stage micrometer scale, which is in millimeter. So we can find out the length of objective through these scales. But in case of stereoscopic microscopes or binocular microscopes, there are two eyepieces. One of these eyepieces is having fixed focus and the other is adjustable to accommodate small differences between the two eyes. In case of trinocular microscopes, the third tube is there to which camera or computer monitors may be fitted to take the photograph of the object. The focusing then, so this is all about the parts and their function. Now let us come to the focusing mechanism. The focusing of the object on the stage is done by raising or lowering the microscope tube or that of the stage through coarse adjustment knob and by using the fine adjustment knob. There are two knobs by which the microscope tube is being raised up and down, or stage can be made up and down. When the high power objective is used, the free working distance becomes very small. In that case, care should be given while lowering the microscope tube so that the tip of the objective lens is not smashed during the process of focusing the object on the stage. Then what is the direction convention? Direction convention is that compass directions are routinely used to describe orientation in the microscope image. Top side is always north, then south, then this is east and west. So north is the top, south is the bottom, and east and west are the right and left respectively. The 45 position is, this is 45 degree position. So it will be northeast, southwest, or northwest, southeast. So this is a direction convention. This is all about the petrological microscope, which are being used to study the rocks and minerals under microscope, and for identification of, so this is refractive microscope, because refraction takes place. Light passes through the mineral on the stage, comes up, and is either it is observed in plane polarized light or in crossed nickel condition. And finally, if we use the conoscopic arrangement to focus the mineral on the stage through conoscopic arrangement, in that case cones of light rays are supplied into the stage of the microscope, and a figure is being produced which is interference figure, and that figure is mostly used to study the optical sign of the minerals, whether it is positive or negative, whether it is uniaxial positive, uniaxial negative, like that, or biaxial positive, biaxial negative, by the figures. So figures are obtained through conoscopic arrangement. So this is all about the petrological microscope or polariscope or refractive microscope, how it is being used, what are its parts, and what is their function, that I discussed. So this is all about the petrological microscope. Thank you.